Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the webinar section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce you to our speaker, Nejc Pavlin, PhD, Project Manager, Process Analytics Development from Sartorius BIA Separations.
Hello everybody. Thank you, Alex, for a really nice introduction. Today's webinar will be about our chromatography solution for analyzing lipid nanoparticles. We recently published this analytical method in the article, today's webinar will go through how this method works, everything what we can see. We will discuss everything what we can see using this method, and so on. But before I go to the main topic of today's webinar, I just want to say a few words about our company, Sartorius BIA Separation. Sartorius BIA Separation is a leading producer of chromatographic monolith chromatographic media. Our portfolio can be divided in two major fields or parts. First is preparative chromatography, where we can offer you simultaneous preparative columns for purification of different biomolecules. These monolithic columns comes in scalable formats from 1 milliliter to 40 liter. They are ready to use, and what is really important, you can use it in GMP environment. Then we have screening chromatography products such as CIMIC disc and CIM multi-well plates. These are products really meant for high throughput purification and cost-effective screening. The second part of our portfolio, and this will be really the focus of this webinar, is analytical chromatography. Here we can offer you analytical columns, which are specially designed to have high resolution, short analysis time, and no carryover. And this column are specialized for individual biomolecule. The second product from analytical part of the portfolio is our PodFix liquid chromatography system, which can be used for process control and product release quality control. This system comes with preloaded and pre-validated methods, reference standards, and much more. Okay, let's go now to the main topic of today's webinar, and this is analysis of lipid nanoparticle. I will start this webinar with this schematic presentation of the workflow in which lipid nanoparticles are produced. Usually, this typical process starts with E. coli production, in which plasmid DNA is produced. For alkaline lysis of these cells to release the plasmid, we offer you alkalizator. This is inline continuous lysis machine, and this machine was produced quite lately. Then in all this workflow, we offers you different purification packages, let's say, simultaneous DIE for pDNA purification, then different chromatographic solution for mRNA purification, and at the end, chromatographic solution for LNP purification using simultaneous OH column. All these steps in this production workflow can be nicely controlled using our PodFix liquid chromatography system, For this system, we developed different platforms dedicated for different parts or for different biomolecules. In this way, we produce pDNA. We develop pDNA platform, mRNA platform, and at the end, now lately, LNP platform, which is specially designed for analysis of lipid nanoparticles. On this scheme here, you can see that this PATFIX LNP analytical platform is composed of two different methods. One method is, we call it LNP switcher, and this two-dimensional chromatography method, and can be used for determination of encapsulation efficiency, size, and nucleic acid integrity. There we have also another method, which is a conventional one-dimensional chromatography method, and is used for lipid composition analysis. My today's webinar will focus on LNP switcher, and yeah, we will go through all these parameters, and I will show you how this machine is used and which data can you obtain from your LNP formulation. So first, I will start with just a short description of the working principle of this machine. As I already mentioned, this is a two-dimensional system. In this system configuration, we use two different column chemistries. First column is a CMAC-OH column and is used for separation of lipid nanoparticles from free nucleic acids. This is a hydrophobic interaction column with large 6 micrometer channels. These large channels are very important, especially when we work with LNPs. LNPs are usually quite big molecules or quite big species, which are very sensitive to shear stress. That's why these large channels really enable us to avoid the shear stress usually present on the columns. The second column we use in this system configuration is our CMAC SDVB column. This column is used for separation and quantification of free and encapsulated nucleic acids. This is a reverse phase column, and we use it in combination with ion pairing reagent. This system is also equipped with two detectors. First detector we use is Mouse detector. This detector is capable of measuring 9 different light scatter angles and is used for determination of particle size or the size of LNPs. And then we have the second detector, UV visible detector, which is used for determination of nucleic acids. What is also important here is that there is no need for any sample pretreatment. The sample is just directly injected on the system, and in the first part, lipid nanoparticles are catched on OH column. Free nucleic acid travel to the second column, where we analyze it using UV visible detector. In between, the particles stay binded to the first column. Then we elute the particles from OH column. The particles travel through the Mouse detector, where we determine the size of the particles, and they get catched on the second column as the VB column. When we open the particles with increasing amount of acetonitrile and very strong interaction with hydrophobic columns, this causes that the particles are opened and the cargo is released. And then we use the same method as before. We analyzed everything what was inside of the particles, and we end up with the chromatogram shown here in the picture. In the upper part of the chromatogram, there is a UV signal where you can see a peak representing free mRNA and encapsulated mRNA. And in the lower part of the chromatogram, you can see the Mouse detector signal, where elution of the particles is visible as a chromatographic peak. Okay, let's go now to the individual parameters which can be determined using this system setup. First, I want to talk about encapsulation efficiency. So this machine or this system can be used orthogonally to the usually used technique for encapsulation efficiency determination, and that's ribogreen assay. When we have particles with just one encapsulated cargo, the results obtained on PATFIX LNP switcher or ribogreen assay gives similar result. But the problem using ribogreen happens because when we work with multiple cargo particles, in this way, ribogreen assay will give you just the total amount of encapsulation. But when we working with or we using our PATFIX LNP switcher, we can nicely determine the encapsulation efficiency for individual encapsulated cargo. So here I have one example chromatogram of the particles in which mRNA and DNA were co-encapsulated. We can see that we can nicely separate both species between each other, and in this way we can nicely determine encapsulation efficiency for individual cargo. Besides encapsulation efficiency, you can see that we can detect lipid adducts also present in these LNP samples. I will talk about that in my next slides. And besides this, we can also determine the size of particles. I will also focus in on this a little bit later. So yeah, of course, a lot of question was asked if we are sure that this machine really gives us the right information, especially about encapsulated mRNA. The question was if all these determined encapsulated mRNA seen on this assay is really encapsulated, is really protected, for example, from ribonuclease decay. So yeah, we performed the experiment. We first record untreated LNPs with untreated with ribonuclease. I think here untreated without being exposed to ribonuclease, and we get the chromatogram shown on the upper part of this picture. Then the same sample was treated with benzonase, and as you can see, free mRNA was degraded, but while everything what was encapsulated is it's still protected. So yeah, the amount of determined encapsulated mRNA was the same in both cases. We were also asking ourselves if there is some mRNA binding on the surface of lipid nanoparticles, and this mRNA was shown as encapsulated mRNA. We tested this with incubation of LNPs with fluorescence mRNA and tried to detect this surface fluorescent mRNA on the surface of the particles using this fluorescence detector. And also here, this result shows that no mRNA was present on the surface, no fluorescent mRNA was present on the surface of the particles. So also this confirmed that mRNA does not bind to the surface of the LNPs using this system configuration and the conditions in which particles are loaded on the system. Okay, let's go now to the second parameter regarding LNP switcher, and this is size determination. So in the same assay in which you can determine encapsulation efficiency, you can also determine simultaneously the size of the particles. As I mentioned before, our mass detector is capable of measuring 9 different light scatter angles at the same time. If this data is then introduced into the mathematical model, we can nicely calculate the size of the particles which are flowing through the mouse detector. You can see in this case the size or the radius of the particles was determined by LNP switcher was around 82 nanometers. And when we measure the same sample on NTA, we can see that we determine 77 nanometers. So both the size results of these two techniques are in very good agreement. But we can also see that we have some separation of the particles by size using this OH column. We can see that when we move to the right of the elution peak, the size of the particles which flowing through the detector is bigger. The particles getting bigger, and also this is nicely confirmed on NTA analysis, where we can see some population of the particles with bigger size than 77 nanometers. Of course, then we were interested in if the binding and eluting the particles from OH column impact the LNP particle size. So what we do, we measure the size of the crude sample of LNPs directly from the machine. The size of these particles was around 132 nanometers. Then the same particles were loaded and eluted from OH column, and then we check for the size of these particles again using the same technique. And as you can see here in the pictures below, the size does not change. The size of the particle was not changed when binding and eluting the particles from OH column. So this is one proof that OH column does not impact on the LNP size. Also, what was another thing we were interested about is how acetonitrile influence the LNP size. So we expose LNP sample to different concentration of acetonitrile. And we can see here that using 7% acetonitrile does not influence the size of the particles. For successful disruption of the particle, at least 50% of acetonitrile should be used. Also these conditions which are used on LNP switcher does not influence the particle size at the end. Okay, let's go now to the third parameter, which can be determined using our LNP switcher machine, and this is a payload integrity. There are at least two processes involved in the degradation of mRNA in LNPs. The first process is mRNA fragmentation, which can happen due to heat stress, shear stress, and cause that mRNA decay into the shorter fragments. Usually, capillary electrophoresis is used for fragmentation determination of mRNA, but recently we published an article in which we use reverse phase chromatography for determination of the mRNA fragmentation. So how we do it, we measure the normal UV chromatogram of mRNA molecule, and then we perform the first derivative of this UV signal. And if we integrate the peak at the highest point of this first derivative and split the peak using this, everything what is on the left represent fragments or shortened pieces of mRNA, and everything what is on the right represent the intact mRNA. We compare these results obtained using this technique with capillary electrophoresis results, and you can see that the results are the same. So this confirms that this technique can be used for the determination of mRNA fragmentation. The second process involved in degradation of mRNA in LNPs is formation of mRNA lipid adducts. I will not go to the really presentation or discussion how these adducts are formed, but I just want to emphasize here that these adducts heavily lowers the activity of LNP formulation, and they can cause different side effects of these vaccines. That's why it's really important that we have analytical techniques which can detect these adducts in the LNP formulation. So how we do it? Here on the upper right part, I have a typical chromatogram I already show you. And if we zoom in this part where encapsulated mRNA is eluted, we can see that beside intact mRNA, showing here in the blue, we can determine the fragments of encapsulated mRNA, so shorter fragments of mRNA, and also the adducts shown here on peak 3, which elute at higher concentration of acetonitrile because they are much more hydrophobic. So in the same assay, besides the amount of intact mRNA, we can determine the amount of fragments and lipid mRNA lipid adducts which are present in this LNP formulation. So here I have one interesting comparison using LNP switcher and capillary electrophoresis for determination of integrity of the LNP formulation. Really, in upper part we can see that with LNP switcher we can determine the amount of free mRNA, truncated mRNA, intact mRNA, and mRNA lipid adducts. But when capillary electrophoresis is used, first we need to open the particles, so we will lose information about free mRNA inside of the particles. And capillary electrophoresis is also not capable of detecting mRNA lipid adducts because this lipid, which are bound on mRNA molecule, does not change the size of the mRNA so much that capillary electrophoresis will be capable of separating this from intact mRNA. And these problems of capillary electrophoresis resulted usually in overestimation of the intact mRNA present in the LNPs. So in this case, around 75% of intact mRNA was determined using capillary electrophoresis, but when the same sample was analyzed using LNP switcher, the amount of intact mRNA was only 52%. So again, another evidence that using reverse phase chromatography is crucial to really have insight what's going on with your payload inside of LNP particles. Here I have another interesting example how this method can be used, for example, for thermal stability assessment of your LNP formulation. So what we do, we aged LNP sample at elevated temperature for 6 days. The temperature was 35 degrees Celsius, and then we sample this sample and analyze it using this LNP switcher, and we can see, for example, at the day 0, for example, we have some amount of non-encapsulated mRNA and some amount of encapsulated mRNA and some small amount of adducts. After 2 days at elevated temperature, all of the free mRNA was degraded. You can see here as fronting, see a fronting of this peak at non-encapsulated mRNA. The amount of encapsulated mRNA really drops. So this is indication that some particles was opened because of the higher temperature, and also the amount of adducts is increased. And everything is even more evident at day 6 Again, very strong degradation observed in non-encapsulated mRNA. The amount of encapsulated mRNA is even lower than after day 2, and the amount of adduct stays the same. So this is another example how this method or this system can be used, for example, for thermal stability assessment of your LNP formulations. Okay, and this brings me to the end of the webinar. Just few take-home message for the end. So, using this LNP switcher, there is no need for any sample pretreatment. The LNPs can be directly injected on the system and then analyzed. This system simultaneously opens the particle on the system, and in this way we can determine the encapsulation efficiency of individual or also with multi-cargo LNPs. We can quantify, of course, the amount of our payload. We can determine the size and size distribution. And of course, we also can gain some information about mRNA integrity, and all this can be done in just one analytical assay. At the end, I want to say thanks to all the people who work and who help to develop this LNP switcher. Especially the people from PC4 department at Sartorius BIA Separations, and of course also all the other people from different departments of our company. Of course, at the end, I want to thank you for your attention, and I will be more than happy to answer all of your questions. Thank you.
Okay, great. Just before we get to the Q&A session, we do have an audience, or a question for the audience from Sartorius BIA Separations. You should see it appear on your screen now. It reads, "Thank you for joining our webinar. We would love to support your next steps. Please choose how we can assist you." So if you could just take a few moments here to answer this question from Sartorius BIA, that would be greatly appreciated. It will remain on your screen for a few more minutes here, so if you don't answer it right away, you'll have the opportunity to do so throughout the rest of the webinar. So we'll just give a few moments, see some answers coming in, so we appreciate it, and then we will jump into the Q&A. All right, so now we'll jump into the Q&A. And as a reminder to everyone, you can go ahead and type in your questions for Nate now. So our first question is, how big nucleic acids can be analyzed using this analytical setup?
Yeah, thank you for the question. There is really no limit how big a nucleic acid, encapsulated nucleic acid, can be analyzed really using this system. We try this system with really small mRNA molecules such as guide RNA, for example, signal RNAs, which are few hundreds or even less nucleotide big molecules. And we test this system with really big mRNA molecules, such as self-amplifying mRNAs, which are usually bigger than 10 kb. There is really no limitation regarding size of the nucleic acid cargos to be analyzed using this system.
All right, and our next question is, can you differentiate between full and empty LNPs using this analytical setup?
Not yet. Using this system, we can really only see the bulk, the LNP sample as a bulk, So yeah, we can quantify how much mRNA is present in the whole sample, but yeah, we cannot distinguish between empty and full LNPs using this method. Probably some different approaches would be needed to try to find this. Probably some ion exchange chromatography or something like that. Yeah, we're working on it, but I cannot disclose much more than I did.
All right. Well, it looks like we are running a bit short on time. So if we don't get to your questions, we'll be passing them along to Nate, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, can you further explain how the disruption of particles occurs on an SDVB column?
Yeah, very good question. Yeah, we were quite hard to find explanation what really going on SDVB column when the particles are loaded. We think that at least two different processes are simultaneously happening when particles are binded on the SDVB column. First reason why the particles get disrupted is because of the increased acetonitrile concentration. The SDVB is reverse phase column, and dilution from this column happens because of increase of concentration of organic solvent. So this is one process involved in opening the particles. The second process is really a strong interaction of particles with SDVB column. SDVB column is really strongly hydrophobic, and this strong hydrophobicity of the column really attracts the lipids which are on the outer side of the particle so much that the particle is just deformed, and in this way we assume that in this way payload is released from the particles, and this enables us that we can further analyze the encapsulated cargo at the end.
All right. Well, thank you so much, Nate, for your time and insights today. It's greatly appreciated.
Yeah, thank you. Thank you very much.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.